Hello, welcome to the BioFAIR Community Podcast. I'm Sam Haynes, Senior Bioinformatician at the Quadram Institute and BioFAIR Fellow. For those unfamiliar with BioFAIR, it is the UKRI-funded initiative to foster a culture change in the UK's life sciences research data management practices by providing end-to-end fair research data management and analysis capabilities, along with the necessary support and training for UK researchers working in the life sciences. This will be achieved through three interconnected commons: the Methods Commons, the Data Commons, and the People's Commons. These will ensure UK life scientists have the tools, resources, and expertise to share their research for others to utilise. In our very first episode, we will be showcasing the flagship programme for the People's Commons, called the BioFAIR Fellowship. Last October, BioFAIR selected 10 researchers and professionals from across the UK to lead the way in embedding fair data and workflow practices across the life sciences. The fellowship, for which I am a lucky recipient, covers 20% of a fellow's time for a year to allow them to work on fair projects. For the next few months, I'll be interviewing the other 9 fellows to see what they have planned and ask how is it going. Without further ado, let's start meeting my colleagues. For this episode, I've managed to track down 3 of my fellows. The very first is Marisa, a PhD student at the Open University who is working on developing training materials to help users follow best practices in sharing their data analysis pipelines created on the interactive bioinformatics analysis platform called Galaxy. Hello Marisa, welcome to the podcast.
Hello.
How are we today?
I'm very well, thank you. I'm excited to be the first person on the podcast, so we can both be equally nervous.
How would you like to introduce yourself to the viewers?
Hi, my name is Marisa and I am in the final year of my PhD at the Open University, where I've been reanalysing public single-cell omics datasets to try and learn new things about how they change in conditions like heart disease and cancer.
How is the PhD going?
I've been really enjoying it. I always think I'm a terrible PhD student because I have too much fun. So like everyone else is kind of running around feeling really stressed, and I'm like, oh, this is super cool. Like I did this new thing with my this tool, and I was reading this article, and going to conferences. So I've found it to be a really enjoyable experience.
Amazing. And what about working for the Open University? How's that like?
It's very interesting because we actually do have a physical campus here in Milton Keynes, which not that many people know about because we're primarily sort of known as a distance learning institution. So all the undergraduates are distance learning, but on our campus we have kind of a small community of PhD students as well as the staff. So it's kind of an unusual university to be a PhD student at, but it kind of has its own benefits because you do a lot of online teaching, which is obviously quite popular nowadays. So it's, they're kind of the experts in that, and we have a nice quiet campus when you're writing up your thesis. That's sometimes preferable.
And how did you find this PhD program? How did you end up?
So I'm kind of an unusual PhD student because I'm kind of older than most, and I kind of came to it as like a later kind of career change. So during COVID, I was really enjoying learning bioinformatics online just for fun, really, as like something to do while I was stuck at home. And I kind of enjoyed it so much that I started thinking, can I do more of this? And then I happened to see this PhD project advertised. The project was already kind of proposed by my supervisor, so it was a ready-made project, and it had so many elements in it that I found really interesting. So like the reuse of public data, and you're kind of working with workflows, which is a kind of a cool way to do your analysis, and looking at kind of single cell data was really intriguing to me as well. So I was like, well, I'll try and apply for it and see what happens. And then I immediately got it. So it was, very lucky that it kind of happened at exactly the right time for me.
Amazing. And a lot of the work that you're doing is on Galaxy, is that right?
Yes, yeah. So Galaxy is this free online workflow manager where you can run bioinformatics analyses and kind of various other types of analyses. Sort of even if you don't know how to do any programming, you can use Galaxy. So it's kind of very accessible to all kinds of users. You can create your workflows there, and my supervisor was a very keen member of the Galaxy community, so she pointed me in that direction when I started, and I found it to be a really convenient way of running my analyses.
To actually use Galaxy, is that based at the Open University, or is it more broader than that?
Well, so because I'm at the Open University, we don't have our own massive computing resources. We're not one of these universities that has their own like high performance computer. So some universities I think do have their own Galaxy server, but I actually use the public Galaxy server. So Galaxy is this worldwide community, and I use the EU server, which is based in Germany. So this is a free server that anyone can access through the internet. So I can run my analyses without having to worry about, oh, my laptop can't handle this amount of data, because it's on their servers, which is great, because I can set something running and then turn my laptop off for the day and come back and hopefully find that everything has finished running the next day. So yeah, there's various Galaxy servers. There's like a US one, an Australian one, and they're all connected in this big Galaxy community, but they're all kind of run separately. And then you have individual universities might have their own private Galaxy server.
And a little birdie told me that you've recently stepped up and took a bigger role in the Galaxy community. Is that right?
Yes. So I became the chair of the Galaxy Community Board, which is a board that kind of meets every few months, and it brings together the leaders of the Galaxy communities. So I'm part of the Galaxy Single Cell and Spatial Omics community. We also have all sorts of different communities that are kind of interested in different fields. We bring their leaders together and kind of get them to swap resources and tips with each other, and then I represent the community board at the Galaxy Executive Board. So I kind of try and share what users are looking for with the people who are kind of more involved in actually keeping Galaxy running, and with the technical people who can perhaps implement some of our wish list.
Wow, not bad for a PhD student, eh?
Yes, it's definitely not what I expected to be doing in the final year of my PhD. I thought it would just be completely writing up, but instead it's community representation and doing the BioFAIR fellowship and all of this. So it's very exciting.
Yeah, and this is an opportune moment to talk about the main topic of this podcast, which is you're a BioFAIR fellow.
Yes, I am, which is great, because everywhere I go, every conference that I go to, people seem to know what BioFAIR is, and they're always very excited to hear that I'm one of the fellows.
Should we start off with what brought you to FAIR, and then we can lead to what brought you to BioFAIR? So, FAIR, what is it, and what does it mean to you?
So FAIR is all about making the resources that you share as a researcher findable, accessible, interoperable, and reusable. And for me, the key thing there is that they are reusable, because during my PhD, I've been reanalyzing public data sets. So I'm kind of very familiar with how different data sets look when you actually try and reuse them, and the kind of the differences in quality between them, particularly in terms of how well organized they are, how much information people have given you in the metadata about what's in their data sets. It makes a big difference if the data set is actually shared in a fair manner, because I think often we focus with FAIR on the people who are creating these resources, whether they're kind of data sets or software or workflows. We ask people like, are you making them fair? Are you doing this and that to make sure that they are available for people? But we kind of forget that the ultimate aim of that is that when we're making something reusable, someone could actually be reusing it. So I'm kind of coming at it from the other end of, well, let's focus on the people who are reusing these resources, and how can we kind of make sure that they are actually doing that, so that all this effort that we're putting in is worthwhile, and how can we make sure that the kind of experience of reusing workflows is as easy as possible for them.
So yeah, you mentioned the sort of two aspects that we're interested in, the FAIR data, so the data itself is findable, accessible and reusable.
And interoperable.
And interoperable. I was thinking, what's the I? The I is interoperable. But what does that mean for workflows?
So I feel like anything that you share as a researcher that is potentially reusable can be shared in a fair manner, really. And I think for workflows, the idea that they should be reusable if you're sharing them makes a lot of sense, because it is a method that you can apply to loads of different data sets. So when I create a workflow as an individual researcher, I'm probably not using it just once. I'm going to run it multiple times, maybe with different parameters or on different data sets. And if I'm sharing it, that means that other people could be doing the same. So I need to make sure that it is actually going to be understandable for them, and that if I'm sharing this method and saying this worked great for me, like it's so amazing, what a lovely workflow I made, that actually it is something that other people can kind of get hold of and run on a different data set. Because otherwise, why am I going to the effort of sharing it in a reusable format? I could just as well write up my method section in my thesis or in a publication and say, well, that's all you need to know, because you're never actually going to repeat this.
Yeah, that makes sense. So this brings us to the BioFAIR Fellowship. How did you hear about it, and why did you apply to it?
So I first heard about the BioFAIR Fellowship from my supervisor, Wendy Bacon, who is the Galaxy advocate who kind of brought me into this PhD. And she kind of had heard about it, and she thought, oh, actually, Marisa, you should probably apply for this. And then I was like, oh, well, it's not for PhD students, it's not for me. And she actually, I think, went to one of the webinars and heard someone saying, actually, yeah, PhD students can apply. So she prodded me to kind of go to the next webinar and learn more about it. And I kind of, for me, the BioFAIR Fellowship this year, with its focus on creating and reusing workflows, it really fitted in with everything that I've been doing during my PhD. So my work with Galaxy, where I'm developing workflows and creating training materials for other people to reuse them, and also just my interest in FAIR from having been on kind of the receiving end of a lot of FAIR data sets and not so FAIR data sets. That was something that I really was interested in kind of promoting and trying to educate people about.
So yeah, what exactly have you proposed for your fellowship? What was in your application?
So there are a couple of main components to my BioFAIR Fellowship. So first of all, the fellowship is kind of giving me time to develop some of the workflows that I've been using in my PhD into more reusable forms, so making them easier for other people to apply to different data sets rather than being set up just for the ones that I'm analysing. So I'm getting an extra kind of day a week over this year where I can make sure that those that my workflows are reusable, and then write tutorials for the Galaxy Training Network, which can kind of guide people through the reuse of these workflows. And the second component of my fellowship is that I am planning a training course this summer. So this will be an online course. I'm aiming for it to be kind of, I'm not sure quite what to call it, whether it's like a seminar or a workshop, but I want it to be over several weeks where people can kind of come and chat about the tutorials that they've worked through in their own time. So they would go to the Galaxy Training Network, we'd all work through a particular tutorial that week and then meet up at the end of it to discuss it. So they can ask questions, they can provide feedback on what was working well for them, whether there was something that they would improve on the tutorials, and we can just ensure that we're all moving through the the training materials and getting to know the workflows. And then towards the end of the course, I want to hold a bring your own data event where people can actually start applying these workflows to their own data sets, whether that's something that they've created in the lab or it's just a public data set that they're interested in learning more about. So I want to kind of take them through what can be quite a difficult leap, I think, in learning bioinformatics, from I'm following a tutorial and I have this lovely data set that's all like nice and prepared for me, everything works wonderfully and I get the results, through to now I'm actually going to apply what I've learned to a different data set. And I think that's where a lot of people get scared by bioinformatics, because suddenly things don't work quite as planned, there's a problem with the data set, or something has to be changed in the parameters to work for their new research question. So I think kind of having that little extra bit of help to make that transition can be it can be a big confidence boost, and it can also kind of provide you with that little bit of practical help that you might need, particularly if you are based at a university like mine where there isn't already a big bioinformatics support service. So I know kind of where you are, there's much more help on hand. If you are developing a workflow, if you're trying to run an analysis and you have problems, someone can be like, oh, you should do it this way, here's some tools you can use. But I think for me at my university, it was very much at the beginning of the PhD, it was like, here's a laptop, get on with it. So I was kind of by myself, like using all these online resources, and for me, I quite enjoy working out how to do things myself, but it does kind of take more time. So I think having that little helping hand can be really useful for people, particularly if they are like a wet lab biologist at heart, because if you love lab stuff, sometimes you're not that fond of troubleshooting in bioinformatics. It's kind of a different mindset, and I think just kind of helping people make that jump to like, actually, yeah, I can do this, and it's not as scary as it seems.
So if our listeners want to join your seminar series, how do they find out about it?
So at the moment I'm still in the planning phase, and I'm still very much learning all of the things that the people whose courses I've taken had to do to create them for me, with all the how do you set up your feedback surveys, how do you like organise the technology. But I'm hoping that this course will be running in June this year. So if someone is interested in learning how to use workflows, particularly for single cell analysis, then they should be able to find out once the registration is open through either kind of the BioFAIR communication channels, so they're like LinkedIn or social pages, or through the Galaxy platforms events page.
Thank you very much. If someone is trying to do asynchronously and they're still playing around with Galaxy, how do workflows actually work on Galaxy? How do you find them, and how do you go about sharing them?
Yes, so there are various places where you can find workflows that will work on Galaxy. So on the Galaxy platform itself, there is a directory of public workflows, so you can kind of find that through the Galaxy interface. There's also the IWC, which I think is the International Workflow Commission, which sounds very fancy, but it's basically a Galaxy repository of workflows. And a lot of Galaxy workflows are also shared through Workflow Hub. So if you find a Galaxy workflow on Workflow Hub, there will often be a little button on the page which you can press, and it will immediately take you into Galaxy and set up that workflow for you, so that you can then feed in your data set and run it very quickly.
So that's feeding in, and I assume the opposite is true in terms of if you have a workflow like the ones that you have and the ones that you're verifying, the reverse is possible?
Yes, so you can export your workflow that you've created in Galaxy, which is quite an easy process. You can actually drag and drop tools into your workflow and link them together and specify your parameters without having to do any coding. So if you're then like, other people will love this workflow, I want them to reuse it, you can then export that workflow very easily to Workflow Hub. You can export it in other forms as well, like in an RO crate and things like this, which are kind of you can then share in different locations. So the way that I'm sharing the tutorials that I'm kind of developing for my BioFAIR fellowship is very easy, because when you actually write a tutorial explaining how to use your workflow, you post that on the Galaxy Training Network, and it automatically, the workflow will get sent through to Workflow Hub. So there's kind of a good interaction between Galaxy and Workflow Hub that kind of ensures that our workflows are getting shared and hopefully being visible and available for people to reuse.
Perfect. Thank you so much. So we've had a lovely overview of your BioFAIR fellowship. Are there any final points you want to start to advertise?
So the timing of my training course in June is set to follow on from the Galaxy Training Academy, which is a global training effort. We have the Galaxy Training Network, which constantly has these tutorials available for you, so anytime during the year you can run through them by yourself. There's this 1 week a year where the Galaxy community makes sure that we're there to support people who are working through these tutorials. So there are kind of Slack channels, there are people online available to answer your questions. So this 1 week this year is happening from May 18th to 22nd. So if you want to do your kind of introductory courses to Galaxy, get to know the platform, or if you're interested in kind of learning about any of the many different topics of analysis that you can do on Galaxy, which includes sort of all sorts of bioinformatics, there's also, you know, climate science, space science, there's a whole lot of different Galaxy communities within our one big Galaxy community. So this 1 week, it will be all about making sure that people have kind of support available when they're running through the training materials. So if you're kind of interested in Galaxy, that is a good time to start learning how to do it. And you can find out about that from the Galaxy website or from the Galaxy Training website.
Perfect. If anybody has any further questions for you, Marisa, how can people get in contact with you?
So you can probably find me on LinkedIn, because I do have a fairly unique name, and you can also contact me through my BioFAIR email address, which is marisa.loach@biofair.uk.
Perfect. Thank you so much for your time, Marisa. It's been really lovely to chat.
Thank you for having me.
What a wonderful energy to start the BioFAIR podcast with. Thanks again, Marisa. Up next, we have Nan, who has just graduated from their PhD in clinical medical research and discusses improving FAIR practices across the UK Dementia Research Institute. Hello, Nan. Welcome to the podcast. How are we?
I'm doing well, thank you. Thank you for having me.
No problem at all. To start off with, how would you like to introduce yourself to the listeners?
Well, I suppose I recently was just awarded my PhD, so I am Dr. Nan Fletcher-Lloyd.
Congratulations.
Thank you.
So now what's next? Where are you based?
So I am a research associate at Imperial College in the Department of Brain Sciences. And I'm also a member of the UK Dementia Research Institute's Care Research and Technology Centre, which is also based at Imperial and also Surrey. But Imperial's kind of where it's all happening for me.
Amazing. How was the Viva defence? Was it okay?
Yes, I really, really enjoyed my Viva defence. I had some wonderful examiners who really made it feel like a discussion about the research rather than really having me feeling like I had to actually be on the defensive or even offensive. Just tell the truth, talk about what you did, what problems you maybe faced, what you did to try and sort of account for those problems, and what did you enjoy about it. It was very it was a really nice experience, actually.
That was really good. And how did you find the actual thesis writing part? Was that okay?
That was okay, because I had 3 main chapters, each of which came from a paper I'd already written, and then it was really just sort of everything that had gone into those papers were in those three main chapters, and around that it was, the usual sort of the introduction, what's the problem, what's my approach, why do I think that's a good approach. So that was sort of the basis of my PhD. So that was sort of fairly easy to write. I won't say it didn't take time, but I knew where I was going with it, so I didn't feel sort of at a loose end.
Amazing. So what exactly was your PhD in?
So I always describe it as machine learning for clinical applications in aging and dementia.
Ooh. And how
Yeah.
did that split into three chapters?
So I had one chapter which was based on essentially creating a model or an algorithm to be able to identify patient-specific anomalous behavior. So when you see someone's in-home daily activity, can we work out when there's a real change in that daily activity? Not necessarily just in frequency, but also in what you're doing. So which parts of the house you're going to. So that's the idea, is that would be maybe indicative of a change in, if not physical health, then potentially mental health or situation. Maybe they have had someone staying with them, something like that. But that can be very relevant for identifying earlier sort of any sort of underlying adverse health events. So like an infection, like a UTI, for example, urinary tract infection. For listeners, I was just thinking, yeah. So that was paper one, which is sort of, that was around supporting people living with dementia in their own homes, so they can stay in their own homes for longer, because we're getting them healthcare a bit more proactively. So that's sort of, we were focused on people living with dementia, or I was, my PhD. But I think that also translates to older adults, regardless of whether you have a diagnosis of dementia. My second paper is using, again, remote monitoring technologies for sleep, nocturnal activity, and physiology. And can we use these technologies in the real world to assess the risk of cognitive decline in older adults? So that was very much aging and dementia. Because a lot of things that we experience as we age can also align with the very earliest sort of, not even symptoms so much as things that just changes that we notice, although I think they are being considered these days more to be symptoms, but changes that we notice in people living with dementia. So that sort of very much combined aging and dementia. And then, yes, the third paper, this is perhaps, it seems more, perhaps slightly more removed from the other two, but it's not. I wanted to see how the approaches, the sort of the machine learning approaches we'd applied, could be used not just on a different sort of data type. So I was actually now looking at, rather than time series data, I was looking at electron microscopy images, but I was also looking in mice rather than humans, because where we get our sort of foundational understanding of dementia often tends to be, at least first, through rodent species, so mice or rats. And can we use some of the same techniques that I sort of applied in my first two works in this work, to try to not just reveal or guide discovery of important aspects of mice brains in relation to, this was more in relation to aging, albeit there is also a link to when is it aging and when is it neurodegeneration, but also to help the scientific process, help researchers. A big part of our work is, can we help or provide a complementary tool or source of information to enhance someone else's sort of field line of work, whether that's research, whether that's clinical decision making. That's kind of what we do as a group, and that was what the focus of my PhD was on.
Amazing. Absolutely fascinating. That sounds like an incredible PhD.
Yeah.
I guess for this particular podcast, we need to return to another aspect of you, which is you're a BioFAIR Fellow.
I am. It's very exciting.
Congratulations. So, starter for 10, what is FAIR and what does FAIR mean to you and your work?
So, yes, for our listeners, findable, accessible, interoperable, and reusable. This is what FAIR means. And generally, I like to think of it as open science and good practice in science, or best practice, which is trying not just to make our science reproducible, but something that not just scientists, but if you want to have a go at home and you've taught yourself and you have that knowledge, the data sets are there, our machine learning models are there, the pipelines are there. You can actually see how we got the results that we got in our papers. And I think the idea as well is not just to build trust, although that is a hugely important part of this process, but it's also to help people sort of understand how decisions are being made in whatever field. Because I think, especially when we're thinking about those from a non-scientific background, all of these decisions are being made based on research, and I imagine it must feel very like, oh, research, research, research. Yes, but what does that mean? What does that look like? So it's not just about building trust, although that is a hugely important part of it, but it's also allowing people to feel, I think, a little bit more in the know and perhaps in control of the decisions that are going on around them, that maybe they are sort of feeling like they should be making, or at least having a voice in themselves. So that's kind of, I think, the end goal of FAIR, at least in my mind. And it also, within the scientific community, I think it's something that has always had the potential to boost collaboration, because you've then got, easier sharing of knowledge. And again, I think as soon as you have more collaboration, I know that there's a saying that too many cooks in the kitchen, but I don't think that necessarily. I think that only applies if everyone's trying to speak over everybody else. And I like to think that FAIR is, hey, I'm just going to leave this here for you. You can take it. You don't have to take it. FAIR is more communicative, I'd say, rather than everybody trying to take the lead. It's more there's room for everyone, and we're all just coming together to try and support each other.
Perfect. Thank you so much for that explanation. I think quite a few people now are broadly familiar with how FAIR is meant to be applied to data, but our fellowship cohort is focusing more on workflows and the sort of analysis of the data and making that FAIR. I just wondered if you could talk a little bit about how it could be applied to your area of research, specifically like the machine learning sort of world, I guess.
Yes, I think it's actually, it's particularly relevant, not just to machine learning, but also when we're thinking, my work is generally with sensitive healthcare data sets. So those data sets, they are coming from data points we have collected from not necessarily living people, but usually living people who are actively participating in one of our clinical studies. So we want them to feel respected. And I think a huge part of that is with our FAIR workflows, that we can there will be different levels that we will break this down. If someone's a fellow machine learning engineer, but just not in this field, we want them to be able to look at our workflow and perhaps understand it at the machine learning level. But we also want someone who is from a non-scientific background, or perhaps even a person living with dementia, one of our people with lived experience. We want to be able to say, okay, this is where we've put not just the data, but what we did with the data, and then the model that we applied to that data. And this is how the model works in terms of its structure or design. And this is how we explain the model's decisions at the end. We want to make that entire process as transparent as possible, and as explainable as possible. And I think that's basically where FAIR workflows come in. I like the idea of workflow because I think it's very much beginning to end. It encapsulates the entire process, not solely just, is the data something that's, I think when we think about data, as important as it is, we're thinking standardization, harmonization, which is a huge part of this, but what about the other parts of the entire process? And I think that's why workflow is very much more my focus, I'd say.
So thank you for this, a general walk through FAIR practices in your discipline. Let me talk a little bit more about the fellowship. What is your plan for the fellowship? What are you proposed?
So I think I mentioned earlier, I am a member of the UK Dementia Research Institute. So my fellowship is two parts. It's contributing to the wider mission of BioFAIR. And the way I'm doing that is starting sort of with what I know and with who I know, which is in the UK Dementia Research Institute, really focusing on understanding and improving FAIR data awareness and practices. And I really want to look at how researchers across different centres and disciplines engage with FAIR principles, what kind of barriers they experience, and what kind of support or infrastructure currently makes things easier to adopt FAIR practices in day-to-day life, what would help in the future, because that's something that I can then bring to BioFAIR as okay, from my side of things in my field, this is what I found out, and how can we bring that together with what you're doing, with what other fellows are doing, what we're finding out, how can we bring that all together and find the common threads among all of this. I think because this is the first time we've had, or BioFAIR has had fellows, we're the inaugural fellows. I think my thing is trying to sort of provide a bit of a foundation in terms of knowledge or research, sort of, that preliminary research that any study needs. I'm trying to do that just from my aspect.
So are you planning any particular projects? What's the day-to-day for you right now?
So we have actually already released a survey, a UK DRI, Dementia Research Institute, FAIR survey. So we're basically asking everyone in the UK DRI about their awareness and their adoption of FAIR practices. And this is from researchers to non-academic support staff. And I designed that with the Associate Director of the UK DRI Informatics, Amanida Sadisa, and also the Early Career Researcher Informatics Committee. So we released that at the beginning of December. So that's already ongoing as a project. And the idea is basically at the end of that, that will be active for 6 months, that survey. And at the end of that, we basically analyse the results and we write a report.
where can people find the survey? Sorry, if they're part of DRI.
So yes, if they are part of the DRI, please, 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 your centre managers are very kindly assisting in disseminating the survey link. So hopefully you should all have that either already or within the next day or two. So please do think about clicking on that. And it's a very quick 10 to 15 minute survey, and even that, it might even be shorter than that, really. And yeah, anything you contribute, it's hopefully going to improve your experience, as well as research and the experience of the wider research community, and maybe hopefully even the impact of our research. So please do at least check that out and consider completing it. Then otherwise, if we're thinking about other things that anyone in the DRI can do, we're also organising a workshop day. So it will be a combination of seminars and a hackathon. So that's just sort of an engagement activity. So please do all get involved with that. More information will follow via your centre managers or UK DRI newsletters. But that's sort of how everyone within the DRI can get involved.
Perfect. And how might the survey influence future decisions? What's the sort of the feedback loop that you're looking into with this data?
I think it will be policy, and I am hoping that from that policy some concrete action, whether it be a tool, whether it be best practice, whether it be perhaps some mandatory training. I know not everyone likes to hear the term mandatory training, but generally the training that I have done, either you should know it already, so it shouldn't take you that long, or you don't know it and it ends up being something that's really interesting and very helpful. So I'm hoping it's kind of a little bit of both when it comes to FAIR awareness.
That totally makes sense. I mean, a lot of grant funding bodies and more and more journals, they're putting the priority on FAIR practices, and this is people's opportunity to actually influence how this is going to be implemented across DRI. So I encourage everyone to engage as best they can. So if people want to find out more about you and your research, how might they be able to engage with you?
So I'm on LinkedIn, Nan Fletcher-Lloyd. Generally, if you search for Nan Fletcher-Lloyd, it's a relatively unique name. You'll probably find me. But yeah, everything. I'm a research associate at Imperial College London. So if you find a Nan Fletcher-Lloyd profile and click on it and it says that, it's probably me. And there are some BioFAIR posts on there, so you should be able to recognise me.
Perfect. And I think, what's your BioFAIR email?
It's nan.fletcher-lloyd at biofair.uk.
Well, I encourage everyone to get involved in the survey, and if anybody has any further questions about how to apply FAIR in either sensitive research environments or machine learning, maybe throw Nan a couple of questions. Thank you very much, Nan.
Thank you.
I really enjoyed that conversation with Nan and could have discussed the applications of FAIR practices to machine learning and sensitive research data for ages. Now, last but not least, we have Chris, a Nextflow aficionado and project lead for the EcoFlow team at UCL. Hello, Chris. Welcome to the podcast.
Hi.
How are we today?
All good. Yeah, how are you going?
Yeah, not too bad myself. How would you like to introduce yourself to the listener today?
So my name's Chris Wyatt and I work at University College London, and my background's more in evolutionary and developmental biology, and I'm completely computational, so I'm not allowed in the wet lab. I broke a pipette once, so we're not going back there. My role is to at UCL is running a group called EcoFlow, and we produce pipelines following FAIR standards for different ecological research groups in the UK. So we go around and we do training, and we also work with specific groups in the UK that want to build using these FAIR standards in using a proper scientific pipeline. And we use Nextflow and NF Core standards for the pipelines that we create. And that's, yeah.
Do you want to talk a little bit about what Nextflow is and a little bit about Nextflow, what NF Core is specifically?
Yeah, so I mean, Nextflow is a bioinformatics pipeline manager, so it helps scientists build proper, decent pipelines that are able to process really high throughput data. They're scalable, so it basically takes advantage of all the new technologies in terms of working with HPCs or any kind of different kind of configuration you might need to do for your pipeline, and makes it a little bit easier. So it tries to break up your pipeline into modular parts, and helps you kind of build your pipeline and then be able to deploy it wherever you need to. So Nextflow has been around for, well, I should know how many years it's been around for, but it's been around for maybe, I shouldn't guess, at least 8 years. But and it's actually started in the university that I did my PhD in Barcelona, in the Centre of Genomic Regulation. So that's where I first came across it, and it seemed like a very small project that helped a few people in a lab, and then they realized how useful this was to actually have a decent framework that any bioinformatician can jump in and use the proper standards to do things. I mean, Galaxy existed before, that many people might know, that has kind of a user interface, and you can fix things together. But this was more for people that are bioinformatics. They're already a bioinformatician in full. Like, they don't need to point and click. They just want to get to the very root of kind of high complexity problems with big data and omics kind of pipelines. So this makes it completely flexible for the user, and that's why the platform really took off. And NF Core on top of that is the actual worldwide community that works with Nextflow. So that's what's, I think, makes Nextflow even more exciting, is that there's this huge community that really care about trying to follow very rigid standards that actually help everybody in the long term. So it creates rules about how you should write pipelines, and they have lots of events. Actually, a little plug, but we're having the NF Core Hackathon, the worldwide hackathon. So they do lots of these events. Our one in London will be on in Stratford, in our East Campus at UCL. So it's a 3-day event where all people come together and we all work on projects that we can all work on together. So instead of also all doing it separately, we're all going to be trying to build modular components that everyone can use in their pipelines. And this is that's what makes Nextflow and NF Core really fantastic, that you have this community that helps build things together. So when you want to build a pipeline using these standards, you can just take it off the shelf from NF Core, because all of that information about every program, a lot of these are already stored into modules, and they can be deployed in any pipeline that you want to use. So it's a really, exciting community. So if anyone's interested in joining a hackathon and interested in Nextflow and NF Core, this is a great event to kind of get experience of how you do this and how you build pipelines in this way.
Amazing. Thank you for a fantastic summary. So you currently work at UCL. You said that you did your PhD in Barcelona. What was your PhD in? How did you end up where you are now?
Yeah, so I mean, I've always been, like I said, very interested in evolutionary biology. And I had the opportunity to start a PhD in the lab of Manuel Orimia in the Centre of Genomic Regulation, as I said, working on actually alternative splicing. So like actually not just what genes being expressed, but which exons are actually being used in a gene to have a different function. So that kind of led me to dealing with single cell developmental data of humans, mice, and cows. But so I had these projects looking at these huge data sets of embryological data from lots of different mammals. And this is the kind of work I actually didn't use FAIR practices, and I didn't use Nextflow at the time. I just did it in the traditional, bioinformatics way, sat down, writing lots of code, putting it all in my paper and saying, goes here, you have to run this script, and then you have to run that script. And it was, you know, a bit of a nightmare because new data sets came out, and then you'd have to redo it again, and it was like, oh no, I've got to somehow repeat what I've done, and finding difficulty actually repeating my own work, let alone trying to repeat other people's work. When, you know, that kind of led me to Nextflow. So that's where I kind of realized that I needed to do things in a more professional way in terms of trying to make my work reproducible, and that's kind of where I came from on that side.
How is your Spanish?
Ah, muy bien. Por supuesto. Después, no, 9 años, no sé. But yeah, so it's okay. I get by.
Amazing.
I'm trying Portuguese now, so it's you know, just to make things more complicated.
Yes, but with Portuguese, you can go to Brazil.
Exactly. No, I love Brazil, but maybe another time. I've done trainings there, so I do for my work. I have had to do trainings in Ribeirão Preto in São Paulo province. So, and there's lots of Nextflow people in Brazil actually. So
Oh, really?
the worldwide community. Yeah. So we have a few people that work directly in that region, and I'm trying to remember. Sorry, off the top of my head. Yeah, so Marcel Ribeiro Dantas is the kind of head of the Brazilian NF Core. So he's kind of heading that kind of part of the world. So there's lots of people around the world that are kind of ambassadors to NF Core. So yeah, it's a good place to. They also do hackathons in Boston, but at the moment, yeah, let's just say the one at the moment is really great because the one in March, it's just you can do it in your local place. You don't have to fly to a distant place, so it's much more environmentally friendly that you can. This hackathon is very local, but they also do a hackathon in Barcelona in September. So they do lots of different events, online or in person, to try to make it as inclusive as possible for people that don't want to travel so far.
Amazing. Thank you so much. So, this is the BioFAIR podcast, and we should be talking a bit more about BioFAIR and FAIR practices. So, a starter for 10, what does FAIR mean, and what does it mean for you?
So, I think FAIR, you know, this is such an important thing to be able to make sure that all your work you're doing is not wasted. And I think what it means to me is that it's a kind of goal that we should be trying to achieve, but maybe not necessarily following every rule. I say that because I think it's a way of trying to, like with my work in particular, I've always wanted to try to improve how I do things so that people can reproduce it. I hate that in the past I've done things in quick ways, because we all have deadlines, right? And we've got bosses pressuring us to make sure that we get work done on time. And that means that you maybe, you know, you could put the data on a different publishing directory that's more findable for people, or it could be that your pipeline, if you made it into an actual pipeline so that people could just run it with one line, people could actually repeat it. And I think it's a kind of, I think FAIR for me is really important, that like let's get together and just talk about how we can do things better, and then try to find practical ways of actually achieving that. Like, not just, I think if we become a big voice box for saying this is how we should do it, and everyone's like, well, we don't have time, then we haven't really achieved anything that's been done before. So I think having a way of giving people practical tips, maybe work cases of how they could improve how they do things. And the same I think for everyone can improve in some way. And I like the fact that FAIR is trying to push that, like that we're all on our different steps of where we can improve our work. And there should be no shame. At any point, you're just like, okay, I'm at this level, great. I could definitely improve how I can do this and be more efficient in making things more accessible and findable and all the rest. So I think that's my perspective on this. Yeah.
Amazing. Thank you for that summary. So yeah, this brings us to the fellowship and the BioFAIR fellowship. So how did you hear about the BioFAIR fellowship, and why did you apply?
I actually can't remember how I heard of it. I think probably someone I heard it through the grapevine from different people. And I was just like, I was interested, I was intrigued of what this would entail, and definitely this is what my work's been trying to gearing up to. Like my current project, EcoFlow, we're trying to move to these better standards. So, but it's within my context, right? It is within ecology and evolutionary biology, but this links to Elixir doing things with lots of other organizations are all pushing for these different tactics, and it's nice to do these things together and try to combine our efforts instead of having them all siloed across different fields. And I think the other question, so where did I find about it and why? What was the second question? Sorry.
Yeah, just what made you apply? But I think you've answered both questions. Don't worry about that.
Okay, cool.
So, what have you planned for your fellowship, or what are you doing for your year?
Yeah, so I mean, I think one of the main things for me is we're able, with the time that this has given me, to produce new training modules for NF Core and Nextflow for early career researchers. So that was the main thrust of my application. To try to, in the past, we've done hackathons and we've done some trainings, but there's always this kind of lag, and there's people that want to use Nextflow and want to use NF Core, but they don't have the confidence to just jump in. And even to join a hackathon, I mean, the hackathon, I said it's for 3 days. You don't have to turn up for 3 days, by the way, if anyone's interested. But people are like, can I just go to a hackathon? Is it, for me, is this the kind of thing I can just go to, and will I learn something? So this is why with the call, we made it really important to have training before, so that people know how to do PRs, pull requests, within the NF Core ecosystem, so that people can go, okay, I want to change this part of a pipeline. Give them the confidence to just try it, do a pull request, and then once they've done that, once you've done this, like it's just those like small beginner early career steps. If you've done those, then you have the confidence to keep going in this direction and building things better in this fair manner. And that's what Nextflow and NF Core is for. Is trying to build these fair standards without having to think about them. So it's a way of baking in these high quality standards, but not using with low kind of effort. But you still have to have that low effort at the beginning, and that kind of, you need to push past that threshold. So we're going to do a training actually on the 5th of March as well, connected to this hackathon, so that people can have an experience of what you're going to do in hackathon, and how, because otherwise people go to hackathons and they just sit there and they're like, oh no, there's people doing stuff, everybody's doing stuff, I'm not doing stuff, I'm scared, or maybe I'm not confident enough to do what I want to do. So that's what this really is pushing on my fellowship, is that we'll be able to do this event and further events in the rest of the year to give as many people as possible kind of experience and training in how they should be working with these technologies.
That sounds really, really interesting. I'm a little bit conscious of the fact that this podcast might not be out by the time these initial events have been planned, but how might people keep in touch and find out about future events?
Yeah, so I mean, the best thing really is the NF Core website itself. Any kind of events they post there, the hackathons. They also do kind of one-off events in cities around the world. So they do which are called Sakara sessions. Sakara is the kind of company that is connected to Nextflow. So they do these sessions where you can hear about other people's work. So these are the kind of really great places just to go in and experience and see what other people in your field are using these technologies for. And yeah, I think that's the main thing for Nextflow. And of course, EcoFlow ourselves, like my initiative, we also have a website where you can see events. Any events that we do are on there. And we try to post anything we can to try to make sure people know all the things connected to Nextflow and of course that are happening in the UK. Yeah.
I have a more general question about how NF Core pipelines are sort of organized, structured, modified. How does that work in practice? Is it sort of democratized? What's the process through, people can issue a suggested change through a pull request? What happens then?
Yeah, exactly. So, I mean, it's all, obviously everything's open. You can see everything that's gone on because it's on GitHub. So everything's ready to go. And on GitHub, if you want to edit, you want to add a new module or you want to add a new feature, you can request it. And a lot of these things, because these pipelines are so public, especially like things like NF-core RNA-seq, which is like a hugely used pipeline. If you put a suggestion or there's something wrong, it doesn't work in your data set, there's people waiting to essentially answer your question. So the community is I can't argue strongly enough how important the community is here, especially on Slack for NF-core and discourse, that you can ask a question and people normally answer within an hour, and they might even add that module for you. People are very happy to jump in and be like, okay, yeah, that's a really good idea. It's going to go on a list of things to do for that pipeline. And either you can do it and you can do a pull request, you fork the pipeline, you make a change, you do a pull request back, and other people are there to review it, make sure it works. It's all got tested. So there's lots of different tests in these pipelines. So as soon as you make a change, these tests are initiated by GitHub Actions so that they can make sure that the changes you make are not going to break the pipeline. So it's very well tested, and then you've got all these different reviewers. So every time you make a change on these pipelines, you just say, reviewer, find someone from NF-core, and more than likely there'll be someone to help you very quickly to make sure that what you wanted to change has worked, or your issue has been resolved. I think that kind of support, just native, by all the community, because it's so big, that's what really makes it, I think. Because I think if you get stuck in bioinformatics, or you need to do something, that pipeline's not doing exactly what I need to do, having these people there available is just enormously helpful to stop you getting these kind of roadblocks, in people's science.
Amazing, thank you. It's nice to have a nice general overview of how NF-core actually functions. So if there is someone out there who wants to learn a little bit more, but maybe wants to learn it asynchronously, what would you suggest their starting point would be to join the community and maybe learn a little bit more? Maybe they need to learn a little bit about how GitHub works. Maybe they need to learn a little bit about how Nextflow works before that.
Yeah, so I think the best thing for this is the Nextflow training, which I initially worked on a lot when Sakara and Nextflow started. It's since evolved massively since I worked on that, and been worked on by too many people to mention. But if you go to the NF core website or the Nextflow website, both websites have access to different training modules. And these are also on videos. So if you are more comfortable going through video training, they have those. And they also have an example. This is what a channel is, say in Nextflow, and then they'll give you examples, and then you can kind of copy and paste text or write your own text within a virtual environment like code spaces. That means that you can have all the tools that you need, and it just gives you a bit of confidence that you can run these things. So there's I could probably, maybe on these, we can link some of these training things in the podcast eventually. But if you just go on to the Nextflow website and just click on community and training, I think it is, you will find all these different training modules, and they're just really helpful. I think it's just useful trying them, going to these talks and on YouTube, just choosing your method of learning basically. And there are events across the UK, especially from us, but also from other groups that do specific early career Nextflow training. So it's always worth listening to, seeing where they are. And on Blue Sky, we also obviously post all these events to try to find as many people across and through our email chains. But it's difficult always to find everyone that might be interested in these events.
Fantastic. Is there any final remarks you want to share with everyone?
No, all good. But thanks for a nice podcast. Thank you for hosting it. It's been great.
No problem at all. And how about if people want to get in contact with you personally?
Yes, I guess the BioFAIR one makes sense, which I think I am.
chris.wyatt@biofair.uk.
Thank you for that. Yeah. So please shout out on there, because we're always, and if there's any projects or you're working with Nextflow or NF-core and you want to build part of this community and you don't know where to look, just shout out to me anytime, and I'm always ready to tell people what's going on, what events are happening. And we can have a mailing list as well. We can add you to the mailing list to make sure that you have access to the events that might be happening near you.
Perfect. Thank you for your time, Chris.
Awesome. Thank you so much.
A great chat to end the podcast with. make sure you look out for more opportunities to engage with the amazing NF Core community. That's the end of this episode. Thanks again to everyone who contributed, and thanks to the UKRI for funding BioFAIR and this initiative. Follow BioFAIR on LinkedIn to get notifications for when the next episode comes out with another 3 fellows. Thanks for listening.